So today, everything we'll be looking at today has to deal with the peculiarities of individual DNAs. Even though everybody will have the same chromosome, uh, our DNA is still very much different. For instance, you as a person, everybody is unique in his or her own way because even though you are inheriting your genetic materials from your parents, uh, your genetic composition is different from that of your dad and that of your mom because you are gaining half of your genetic material from your dad and half of your genetic material from your mom, which means that the combination that you have, even though it's made up of your pay that is gotten from your parents, is entirely different from what they have individually. So and based on the fact that your genetic compositions are different, we are able to capitalize on these individual variations and maximize it. So that is one of the ways through which we capitalize on these variations is what we'll be talking about today. So we'll be starting with DNA profiling. So when we talk about DNA profiling, is more popularly known as DNA fingerprinting. So when we talk about who knows what is a fingerprint? Let's start from there. What do you think a fingerprint is? Anybody, please. But I really wanted to answer me on time so that we can make progress. Anybody can tell me what is a fingerprint? What do you think a fingerprint is? Are you want me to be calling your names? Who has heard the word fingerprint before?
So,
Yes, go for it.
so I think it's a specific biometric that is left on the friction ridges of the human finger.
Yes, that's really good. The reason why we many when somebody is at a crime scene, when you visit a crime scene, and those forensic experts come around, they will now be looking for fingerprints because when they are able to see a fingerprint, they believe that no two persons have identical fingerprints. So if they're able to see a fingerprint at a crime scene and they're able to match it with anybody, it simply means that that person that owns that fingerprint was most likely the perpetrator of the crime or was at the crime scene. So and that is going to lead them to start asking some questions. And the same thing is what we are doing here. But instead of fingerprint, literal fingerprint, what we are studying here is the DNA of a person. Because they know that if somebody leaves a blood sample, for instance, if there was a crime and they are seeing blood at the crime scene, they would be able to identify they'll be able to extract genetic material from that particular blood sample and match it to anybody that if they're able to match that DNA with anybody else, it simply means that that person was probably at the crime scene. So this is not just crime, but these are the ways that will be that this methodology can also be applied. So by definition, DNA fingerprinting or DNA profiling is the process of determining an individual's DNA characteristics. So, like I said, your your DNA, your complete genetic materials are entirely different from mine. So, because and which means what is different in your genetic material from mine are the DNA characteristics. So, if you are able to understand the variations, the differences in our DNA materials, then we are most likely going to be able to see that see what is different and what is peculiar to each of us. So DNA profiling on its own is a forensic technique. Is a forensic technique, which means is an investigational, is a potent crime investigation tool for comparing crime suspects' profiles to DNA evidence so as to assess the likelihood of their involvement in the crime. So it's still the same thing. If there was a blood on this, if something happens and maybe on this curtain they saw splashes of blood because it was left at the crime scene, that is an evidence. So they will now collect that evidence, extract DNA from it, and if we have like 10 people that we were 10 suspects, if you suspected 10 people, we will now figure out if we are sure that that blood is from the suspect, maybe the suspect, or let's say somebody that was shot at the crime scene but managed to run away. So we are going to collect that kind of sample and we are going to extract DNA from it. We are now going to identify suspects. People that we suspected could have been the ones that perpetrated that crime. The people that we suspected could have been the ones that came to rob. So we are now going to match the DNA at the left at the crime scene with the DNA of the suspect. So which means that we are able to assess maybe these persons are more likely to be the ones that perpetrated the crime. But DNA fingerprinting is not just a forensic tool. It is not just a crime investigation tool. It is also being used for parentage testing. For instance, now, if you want to, for paternity disputes, if you want to know whether you are the real parents, you want to confirm whether you are the real father of this baby, this is a method that they use. And there are some countries that when you want to go, when you want to travel to these countries with your kids, if you are married and you have children, they may want to test they want to carry out parentage testing to really affirm that you are the parents of those children. So that, because why is this very, very important? Without establishing parentage testing, without establishing that somebody is actually the real father or the real mother of a child, they can be, a condition, a situation known as child trafficking, in which anybody will be moving children everywhere for slavery purpose. The only way that serious countries can avoid this is to ensure that anybody that is traveling with any child is the real parent of that child. And DNA fingerprinting allows us to do that. There is also something that is called genealogical research. So if you want to know your family tree, who are your ancestors? If you want to know whether this person is connected or related to you, then they also carry out DNA fingerprinting. Let me explain that genealogical research much more further. There is a website that is called ancestry.com. Let me see if I can display it. What it helps is that if you want to know where your family lineage is from, or where your forefathers are from. Okay, I think so. What you can now do is you will now so that they can actually collect samples from you, and they will now look at where you can match it, where it's more likely you have closer relation to it. So this is a website. I don't know. Can you guys see my screen? Hello, can you see my screen?
Yes, sir.
Yes,
Okay,
sir.
great. So the website is ancestry.com, and you can see there is a genealogy, and so you can actually, on this platform, you can get insights from your DNA, whether it's your ethnicity or your health. So the way it works is that they would send you something to collect samples. We are still going to get to the part where you collect your samples, the kinds of samples that they collect and how it's processed. And they already have database of DNA from across the world. If somebody wakes up and says, I want to know my family history, and want to know, let's say a Black American wants to know where his forefathers emigrated from, because Black Americans most likely originated from Africa. Their forefathers came from Africa, but there are many African countries. There is Ghana, there is Sierra Leone, there is Nigeria, there is Rwanda, there is Egypt, so several other African countries. So if a Black American wants to know where his forefathers are from, so you, from the corner of your room, what you just do is that they send a kit to you. They collect some for your buccal smear. You just rub it through your mouth so that they can collect some cells from your mouth. Then they plug in the DNA that is extracted to their database, and where the region where you have the closest match, because you're expected to have something similar to your forefathers than other parts of the world, from where you are from than other parts of the world. So that is how it works. And this platform, and this is not just the only one. There are several of them that have this particular tool that has really, really helped people to know where they are from. So, and there is a vast application of this particular methodology to serve the purpose. So that is what we talk about. So what we are always looking forward to is that which region has the best match with this particular person. So DNA profile too is not just humans that they do it for. You can talk about plants. We can use it for plants. You can use it in agriculture, and also in the field of zoology. It has extensive application. So how do we carry out this DNA profiling? What are the processes that it takes? The first thing is to get the reference sample, which is an individual's DNA. For instance, now we want to collect, this is the reference sample is like, this is a sample we are trying to know its origin. This is the sample that, in the case of somebody that is trying to know where his or our forefathers are from, that person's sample is intended to serve as the reference sample that you're matching. So, and the way it's collected is through a buccal swab. So there is, I think I will still show you a particular video of how it's collected over there. So normally, because the reason why a buccal swab is usually the sample of choice is that you don't need to teach anybody how to collect it. Anybody can collect it. It is not painful. You just insert a swab stick to the corner of your mouth and you roll it, you moisten it. It's either that some cells will have dropped in that buccal swab, then you extract DNA from it. But if for anything the buccal swab is not available, then you can actually get DNA samples from other parts of the body, including the blood. It's also present in the saliva, the semen, lubrication, vaginal fluids, even your toothbrush. If you are using razor blade, if a guy that is always shaving, you can also get some cells from the razor blade. Or we have stored samples. For instance, if somebody has already died, but they collected biopsy tissues, you can still extract DNA from them. And in developed countries that they have sperm banks, DNA samples can still be extracted from that. And furthermore, in cases, for instance, when you are trying to, let's say there was a air crash, there was a plane crash, and the bodies have been, on the plane, we can have hundreds of persons on the plane. And let's say in a fatal air crash, let's say there were 300 people on the plane and everybody died. If you've seen a plane crash before, the plane is usually engulfed in fire. So which means 300 people were burnt beyond recognition. But many countries are still able to identify, even though you cannot physically say that this is Taiwo, this is John, this is Kendi, this is Mustafa. Because even though you are not able to physically say it, DNA profile too allows us to still be able to collect samples from different bodies and match it to their closest relative. For instance, if a father died in an air crash, what they are just going to do is to collect a sample from the child of that particular parent that was involved in that aircraft. Because you are sharing some half of your DNA materials with your father, it simply means that anybody among those corpses that died that has more similarities to you than other persons is most likely to be your dad. So that is how they are able to identify corpses that were born beyond recognition. Even in family lineages, people that they've buried for years, we've had cases of in some very, very rich families in which about 10 years later, somebody now, after a man has died, somebody now arose and said that this person that died was my father. And they were like, no, how is that possible? Then they went to look for the remains of the person that had already been buried. They collected some samples, they extracted DNA from it, and they were able to match it with the person that was claiming that the late was his dad. So when you do that, you are able to profile human remains. You are able to connect and identify where this person was probably from. And when the person that you are also matching, when the mother of that baby is also alive, let's say the dad was the one that was involved in the air crash, but the wife of that person that was late was still alive, and the child too. So since the DNA composition of the child is the combination of the genetic materials from the father and the mother, you've already gotten the mother that is alive. You already have access to half of our genetic composition, and the balance would be seen in the child that's expected to be from the late man. So by the time you compare it, it's like X plus Y. You are looking for X plus Y is equal to Z. But you are now looking for X, which is the father, but you have the result, which is the child. So what you just do is that you subtract the DNA materials of the mother from that of the child. The remaining ones will be that was gotten from the father. I don't know, does that make sense to you guys? Do you understand what I just described? Let me ask.
Yes, sir.
Okay, great. So let me continue then. So that is how DNA profile. So the reference sample is analyzed to create the individual's DNA profile. Then after getting the DNA profile of the reference sample, you are now going to compare it against another sample to determine whether there is a genetic match. So the goal of DNA profiling, the goal of DNA fingerprinting is to establish genetic match, to see which one is the closest relative, which one is more connected or more related to the reference sample. So the closer to the reference sample, the likelihood that these persons or these samples or these sources are related. So the first thing that you do is whichever sample, because you are not collecting the DNA directly, you are collecting something that is expected to contain the DNA. Let me give you guys a quick background. The DNA is stored in the cell. If you remember the cell, very well, if you remember the cell, you will know that the cell has a nucleus. It also has a cytoplasm. So there are many components in the cell that are present in different parts of the cell. But for the genetic material, where do you think, Ayomide Omotolagbe Kehinde, so where do you think the DNA is stored? So if I ask you a question and you don't answer me, it simply means you are not with us. So I can easily kick you out and you are absent. So I'll ask again, Ayomide Omotolagbe Kehinde, are you with us or you just logged on and you are busy doing your stuff? It's obvious that this person is not with us, so I'll kick.
Sir,
So where is the DNA stored? Where is the location of the DNA in the cell? Do you know?
cell nucleus?
Yes, so that is true. The nucleus is where the nucleic acid materials, nucleic acid is present. So it's located in the nucleus. So if we want to get to the DNA, we have to enter the cell and also get to the nucleus. So when we have entered the cell, then we have to rupture the the nuclear membrane. That is what protects the nucleic materials. So we rupture the cell. We also rupture the nucleic acid, the nucleic membrane. And when we've broken the gate, the nuclear membrane is like the gate to the DNA. So when we've broken the gate down, then the components of the nucleus are then released into the container that we can now purify and get our DNA. So that is the underlying principle for DNA extraction. Because like I said, even though you are collecting buccal smear, the blood, those are not the DNA. The DNA, they contain DNA, but they don't contain DNA alone. So before the DNA can be analyzed, it must be extracted from the cells and purified. And there are many ways this can be accomplished. There are many methods. There are several methods of extracting DNA. Like I'm saying, we've had cases in which DNA was extracted from the hair strand. DNA can, like I said, from the saliva, DNA is present there. DNA is also present in the buccal smear because cells are present in these in the buccal smear. You can see cells. In the saliva, you can see cells, and so several other parts of the body you can see cells. But it is not the buccal smear alone. It is not the sputum that is the DNA. The sputum contains cells that will contain DNA. So, and the DNA is our target, not the sputum. So the way through which we get to the DNA is made. There are several approaches, but they all follow the same procedure. So the first thing that you do is that we break up the cell and nuclear membrane. Cell membrane protects everything. But after breaking up the cell membrane, then you still need to break up the nuclear membrane, which contains the DNA. So when we've broken up the cell and nuclear membrane, then DNA is now released into the solution. And once the DNA is free, it can now be separated from other cellular components. So what to do is that the first thing is that we break the cell membrane, we break the nuclear membrane, we release the DNA and other components into the solution. And even though the DNA is now part of a lot of other cellular components, we will now start the process of purification, getting rid of other cellular components so that it is now the DNA that is free, that is available, that is left. So that is the protocol. And there are several methods, and the most common method of DNA extraction include organic extraction, that is called phenol chloroform extraction. We also have chilex extraction and solid phase extraction. But I don't want you guys, since you guys are still in 300 level, I don't want you guys to stress yourself too much about specifics. Just understand what we are doing. That when we are saying we are doing DNA extraction, what we are essentially doing is that we are first breaking the cell membrane, then this is followed by the nuclear membrane, then we now start the process of purification. So we now start the process of purifying the DNA from extra components. So I have a short video here that would explain how we extract the DNA from the buccal smear. And I want you guys to be quite attentive. So for somebody to collect the sample, we are collecting it from the buccal smear. You have to rinse out your mouth. You have to wash your mouth because many things, food particles, you find it in your mouth. Contaminants, you find it in your mouth. So you have to rinse out your mouth twice with water. Then this is like the swab stick. It's just like the regular swab stick, nothing abnormal. So this is the swab stick that they use to collect it. So what they do is that the side of the cheek, it goes into the side of the cheek, then you roll it, you press it firmly against the cheek. So it's touching your cheek and you start rolling it over and over again. So after you've rolled it through your cheek side, then you now can you see there is a tube that the swab stick contains. It's like fluid, and this tube contains solution, DNA extracting solution. So what you now do is that that sample that you've collected in your mouth, you now transfer it to the tube that contains the DNA extraction solution. You'll be rolling it in that tube so that it will be dissolving in the samples will be dissolving in that fluid. So you cannot take it from there. Then, so which means our work is done with the swab stick. That fluid that you've dissolved the component of that swab stick that we are trying to extract the DNA from is what is in this tube. The other thing, the subsequent processes are like we have vortexing. Vortexing is just something that have you guys seen a vortexing machine before? Something that has this kind of head. Have you guys seen it before? Has anybody seen this machine?
No, sir.
No, sir.
Okay, it exists. Okay, maybe when you guys start coming to the lab more frequently, you will see it. But it's something that is everywhere. It's not something that is. What it just does is that it continuously vibrates. Let me see if it. Let's see that. So after vortexing, incubation, so just a series of you had an extraction fluid, you spin it, you discard it in water, you replace it with water, you put it on dry bath and on heat. So this is just the way DNA is work. By the time, like I just wanted you guys to see how it's just you are vortexing, you are placing in dry bath, you are adding fluid, you are removing the supernatant, you are adding new fluid. So that is just DNA extraction is not a big deal. That is just how we do it. And when it's done, because the DNA is very fragile, that's the reason why it's at the end of the whole process, they start placing it in ice. And so that is just what they do. And if you want to do something like so once you've extracted the DNA is what you now use for subsequent purpose. We are still going to come back to this because the video now starts talking about PCR, and I think there is a part of this presentation that has to talk about PCR. So it's not the reason I just wanted to see is that when we say DNA extraction, it's not something that is complex. It's not something that is magical. You are just You are just pipetting, collecting from one container. You will mix, you will vortex, you will discard the supernatant, you replace it with another extraction fluid, you mix again, vortex. So that is it. So at the end of the whole process, the DNA would be present, will be suspended in the extraction fluid, and that is how we'll be using it. And you have the DNA.
Sir,
Any question? Yes.
yes, sir. So I understand that each of the steps play a role. Like maybe one of them is used to break down the cell membrane and the other. But which I don't know which one. Like the vortex. What exactly does the vortexing do, and what exactly does the incubation do in that process?
Okay, very good question. When you are vortexing, the vortex is just mixing. It's serving as a mixer. Hmm? The vortex is serving as to mix, to ensure that the fluid you've added is getting in touch with every cellular component and is able to perform its function. That is a vortex. The incubation tool is giving, for instance, if a reagent needs to work for like 30 minutes, or it needs like 5 minutes to perform its function. So incubation gives it that temperature, that condition. For instance, if an enzyme would only work at 65 degrees, so incubation, if you see the incubation in that video, is at a particular temperature, like let's say 70 degrees centigrade. So it stays for 70 degrees centigrade for like 5 minutes because that temperature is important for it to work, and it has to be for an adequate time. The vortex does not give you that temperature. It's the incubation pad that has that temperature regulation. So, which means that if you are leaving something that ought to work at 70 degrees, if you are leaving it at room temperature, even though you've given it like 5 minutes, it is still not going to be working. It is still not going to perform that function because it needs that temperature requirement. So the incubation medium provides the temperature while the vortexing ensures that it is well mixed. So the stuff that are getting the job done are the reagents that you are adding. And these reagents vary from one method to another. It varies from one method of extraction to another, and it varies from one kit to another. So I think, okay, if I still want I really like the way you asked the question. Maybe let me see if I can quickly get you. Let me get you a sample here so that you understand everything that you'll be using is not you that will be. If you are doing extraction, then you are using a kit, and there are several manufacturers of kits. There is one by Thermo Fisher. So if you are using this one by Thermo Fisher, you are going to buy it. This is how I want to. So the first thing you know if you want to buy an extraction kit is that what is the nature of the sample. If you're extracting from buccal smear, then you need to buy a buccal DNA extraction kit. If it's blood, you buy a blood DNA extraction kit. So if it's buccal DNA, then this is the kit that you buy. So every kit comes like this. See, it contains its own pack. I really want you guys to see so that you understand that everything that we are saying is not magical. It's something you can actually do. So as is number of swab, one swab. So you can just order now. You can order. Depending. So once you've selected the type of material you are using, another thing you select is that how many. Because this one is for 50. If I 50 then of 20 samples, if I just want to use it for 50 samples, this one is for 96, this one is for 100. So then another thing you understand is that every of these kits, they have their own manual. Everything will come with a manual. So it's that manual that will tell you what to do for that particular. I hope this makes sense and even For this particular kit, it actually contains its own protocol video too. They're going to show you how to use it. So you just watch all these videos, but most importantly, it has its own manual. It has its own manual that will be telling you that for 5 minutes, add solution A. What you just need to do is that go to solution A, pipette it, do it. That is all that it means. It's not something that is complex. I hope that answers your question.
Yes, sir. Thank you, sir.
So there is nothing, as you can see, they have, even the kit will have its own printed material that just take to your lab. Okay, they said we should pipette this, pipette the sample, mix it together. That is just it. DNA extraction is not magic. You are just pipetting. As long as you know how to use the pipette, you are just pipetting. I don't know if yeah, I think I'm thinking if I can.
In this video, you will learn how the PureLink XP endotoxin-free maxi-plasmid purification kit enables isolation of large quantities of ultra-pure endotoxin-free plasmid DNA for use in your most sensitive downstream applications, such as transfection in primary, immune, or neuronal cells.
So this is how the kits come. This is how they come. So you just unbox it.
For gene therapy and plasmid DNA vaccine research. This kit is based on novel maxi columns featuring the enhanced anion exchange membranes, allowing vacuum or centrifugation-assisted workflows, which are substantially faster compared to the gravity-based workflows for older resin-based columns. This protocol takes as little as 90 minutes. Prepare 100 milliliters of overnight bacterial culture for plasmid isolation with a measured density at A600 of 2 to 4. To lyse cells from your overnight culture, first harvest cells by centrifugation at 4,000.
This is not even for the normal DNA sample. Well, I'm just trying to tell you that the manual is there. You'll just be collecting it. You'll just be collecting you'll be pipetting, whatever they ask you to do. You just do it. Simple. It's simple and straightforward. So DNA extraction is not magical. It's just the way you conduct your regular tests in the laboratory. So so that is just it. And you can watch videos on YouTube about how to extract DNA using kit. So the kit will come with all the reagents that you need, and it is going to come with an instruction manual on how to go about it. The only thing you need to provide in your laboratory is a vortexing machine and a place to provide that temperature, incubation at a particular temperature. So that is everything that you need for DNA extraction. So let me move forward now. Let me continue now so that we can really finish this on time. So after you've extracted your DNA, the DNA is there, but that is not the end of it. You need to now start looking for the peculiarities. What is unique about that DNA and matching it with the reference sample. So what you are doing for the reference sample, the stuff that was left at the crime scene, you're extracting DNA from it. And from the persons you are suspecting could be the criminals, you also collect samples from them and you extract DNA from it also. Then you will now match. Okay, but after you've extracted DNA, you will still carry out subsequent procedures on it. And the first, you can now choose whichever way you want to proceed. There is a method that is called RFLP, and there is a method that is called PCR analysis. So it is not the DNA. You've already extracted the DNA, but the DNA alone is not what is going to tell you whether these products, whether this DNA are from the same sources. We've only extracted it. So you need some subsequent steps to now tell you to bring out the profile, to build a profile and determine the peculiarities of the DNA from one or similarities from the DNA that you've collected. So the first one is RFLP. RFLP stands for Restriction Fragment Length Polymorphism. And the key word for this method is restriction. Okay, now let me give you a quick background. When we talk about a DNA on its own, if you know the DNA is very long, double-stranded, is very, very long. If we stretch a single DNA, I think it's up to 10 spot fields, 10 spot fields, if you stretch it. So, but what a DNA, in restriction fragment length polymorphism, we make use of enzymes that are called restriction enzymes. A restriction enzyme is able to cut the DNA at a specific location. So there is a specific point that a restriction enzyme can now cut that DNA into tiny pieces. And that is called a DNA fragment that differs in size from individuals. So what you do is that the DNA is very long. So you introduce a restriction enzyme to cut the DNA into small pieces. So after you've generated a DNA fragment, then you are now going to separate the DNA fragment using a process that is called gel electrophoresis. So the generation of the DNA fragments now leads you to a protocol that is called the Southern blot. You guys may not worry too much about knowing the specifics at your level, but I really want you guys to at least have a rough idea of what we are doing and how we go about it. So after the genetic material, the DNA that you've extracted, you introduce a restriction enzyme to cut it into DNA fragments. The DNA fragments, you now use gel electrophoresis to separate it into size. After you've separated it, you carry out a protocol that is called Southern blot. So the simple way to describe Southern blotting is as a laboratory technique that is used to detect a specific DNA sequence in a blood or tissue sample. So a restriction enzyme is used to cut a sample of DNA into fragments that are separated using gel electrophoresis. And the DNA fragments are now transferred out of the gel. The gel electrophoresis will separate the DNA fragments based on their size, and you will now transfer that DNA fragment out of the gel to the surface of a membrane. And when you've done that, you will now expose the membrane to a DNA probe that is labeled with a radioactive or chemical tag. So the way we make the interpretation is that if the probe binds to the membrane, then the probe sequence is present in the sample. So this is a picture that shows you how this works in its entirety. So the first thing is that, don't forget, we said it starts with, if you remember what I said, RFLP starts with the PCR, the genetic material that you've collected. Then the first thing you do to it, the first thing that you do is that, if you still remember my analogy, the PCR, you introduce a restriction enzyme to cut the DNA into tiny pieces, the DNA fragments. So when you've cut the DNA into tiny fragments, you now load it to a gel, electrophoresis gel, that is now going to separate the DNA fragments based on their size. So after you've done that, then you now go to the first step in Southern blotting, which is something that you are looking at on your screen. So what you look at is that the DNA, around that one, is the gel, and so it's those long black lines are the ones that contain the separated DNA materials. So what you are now going to do is that you will now introduce a nitrocellulose filter paper. You attach it to it. From that, what we are trying to do is that the separated genetic material, the separated DNA fragment, we want to transfer it onto this nitrous filter paper. So as you can see now, so you place a nitrocellulose filter paper directly on that gel. Then on this cellulose filter paper, you will still add blotting paper. These ones are going to ensure that the cellulose paper filter is well soaked and is not leaking away. So look at the stack on stage 2. We have the gel, that contains the separated DNA fragments. On top of it, we have the nitrocellulose paper, which is the yellow. Then we also have the blotting paper. The blotting paper is to ensure that this is transferred. Everything is just that you are trying to produce a sandwich. If you have seen burger before, you know, if you have seen a beef burger before, we have bread outside, then you have a layer of cheese, you have a layer of beef, you have a layer of cabbage, tomato, onions, that has that sandwich that is pressed together. So we are trying in stage 2, we are trying to ensure that there is a sandwich, that thing that is at the bottom, we are trying to rub it off, we are trying to transfer it onto the nitrocellulose. Now look at the difference between number 1 and number 3. What was on the electrophoresis gel has now been transferred onto the nitrocellulose filter paper. If you look at the stage 3, what we had on the electrophoresis gel, on the blue, or is this somehow blue pad, has now been transferred onto the nitrocellulose filter paper. So that has been done already. So and we've achieved that. So because it has now been transferred, the reason why we need to transfer it because the gel is solid. You cannot be working with a gel when it's very, very solid. So when we've transferred it, we now look at nucleic acid probes for hybridization. And what this probe does is that, let me describe this using a magnet and pin. If I have, let me see, what can I show you guys that will, okay. I want to describe number 4 now. So what you are looking at in my hand is a container containing sugar. So if I have, do you know these safety pins, very small pins? So if I have pins in this sugar, how do you think I'll be able to safely pick out the pins in this sugar? Somebody should try. How do you think I will be able to remove all the pins in this sugar without injuring myself? Who can go for it?
By using magnet.
Yes, that's very smart. So because I have sugar and the pins are very small, and if I say I should be inserting my hand into this sugar, I'm going to injure myself. And if I'm even able to collect like 2 or 3, I will not be absolutely sure that I've collected everything. But what I can do now is to bring a magnet in contact with this sugar. I can open it like this. Let's say this is my QR charging pad for my phone. But let's say this is a magnet. I can now bring a magnet in contact like this. All the pins would adhere to this magnet. And I can convincingly say that I've already removed all the pins without injuring myself. The same mechanism is being applied here. Let's say we know what we are looking for. We've already transferred, don't forget, the nitrocellulose filter now contains the DNA fragments that has been separated based on their size. And what we want to do is that we want to specifically identify whether there is a match. So what we are now going to do is that we are now using something that is called DNA probe. The DNA probe is serving as a magnet, while the nitrocellulose paper that contains all the genetic materials is serving as a sugar. But in that sugar, we don't know whether the specific DNA, something that can attach, that is specific for that DNA probe, that will tell us whether there is a match or not, whether there is there. So what we do is that we introduce the nuclear probes, DNA probes, that will specify, and when there is a match, when it is present, then you are going to find the DNA probe adhering itself to the regions where it is located. So that is what we do, because the DNA itself is colorless. You cannot specifically see if there is hybridization. So that mechanism is called hybridization. So probes hybridize to complementary DNA sequence. We are, from the sample that we've collected, we'll be able to design a probe. Areas that are peculiar to that particular sample, that is going to serve as a probe. So when that area is present, it is now going to attach to the corresponding area on the nitrocellulose paper. If you look at it, compare what we have in 4 to 5. So we've already, that blue zigzag is a DNA probe that is trying to find areas that is specific. It's trying to locate the pin. It's like the magnet trying to locate the pin in the sugar. If you now go to the stage 5, the areas where you are seeing those blue lines are the areas where the pin is. So the magnet has already attached itself to the pin and is now located where it has found the pin. It is like this is the place where we are seeing matches. This is the place where we are seeing matches, where there is a pin match. So after you've done that, you are now going to print that area out. You are now going to print that hybridization out into what you've seen in 6. And that is going to tell you that you are now going to see a pattern. Now so that is for a patient that you are suspecting. That is the hybridization pattern. Then for another patient, for another suspect, you carry out the same similar procedures. Then you are now going to match this autoradiogram with that of the reference sample. And you now see the one that has the closest pattern is one that we can say is similar. So that is how the RFLP works with southern blotting. I know you guys are at 300 level, but I still believe you can get as much as you can from my explanation. So that is how this works. So that was how things were done in the past. But with the advent of PCR, we've been able to now narrow it down even more specifically and get even much more out of our DNA. Don't forget, the RFLP and southern blotting, they are making use of DNA that you've already extracted. The PCR2 is also making use of DNA. So the full meaning of PCR is polymerase chain reaction. Have you guys been taught PCR? Has anybody taught you PCR? Answer me now. Has anybody taught you PCR?
Yes, Mr. Isaac.
So, at least you guys are by there, but let me still give you practical explanation of what a PCR is. So, PCR2 is also making use of the extracted DNA. So, the PCR, what it does is that it mimics the biological process of DNA replication. DNA replicates. Copies of DNA are produced. More copies of DNA are produced. So, the PCR method is based on this understanding that DNA replicates. But instead of now focusing on the full replication, because the entire genetic material is undergoing replication process. But in this particular, for the DNA, it is the DNA replication, it is confined to a specific DNA sequence of interest. What we are saying is that it's a specific region of the DNA that we will artificially replicate, would stimulate replication. So that what we are looking at is that more copies of a specific region of the DNA is replicated. More copies of a specific region of DNA is replicated. And so with the advent of the PCR, when the PCR emerged, we were able to really do much more with what we've been able to get before. Because what we are looking at is that instead of that particular using the DNA probe method, that area where we want to hybridize, we'll find the DNA that it's representing that. Then we are now going to amplify it, produce more copies of it, if it's present or not. So that is what the DNA, the PCR is telling us. So let me quickly see if I can, the video that I paused the other time, it continued with the PCR. Like as usual, the PCR2 is not a magic. It's something you can quickly get on your own. So, but while, like we say, you need like a probe. Sorry, you need to be able to pipette. As long as you can pipette on your own, you won't have problem with these genetic procedures. The only skill you need is your pipetting skills. So we have, so as you can see, you collect, after you've extracted DNA, you collect the DNA sample, then we also have the reagents for the PCR. So for 10 microliter of DNA, you add 40 microliter of PCR reagent mixture into your tube. So just pipetting. So those small tubes that you are looking at, that she's trying to put the samples in, they are called the PCR tubes. They are the ones that will now go into the PCR machine. So I want to show, okay. So this machine is the PCR machine. So everything that you've put together, the DNA sample plus the PCR mix, you are going to mix them very well. Then you just put it in the PCR. You just put it on the PCR machine. As look at the top, this is that black region. It's also where you arrange your tubes that contain the PCR mix. Now, so if you remember, I said gel now. Remember, so after the PCR, everything has its protocol, it has its duration. It's like a cycle. But for your level, I don't think I would be going into details. So that thing that you mixed, you will not know what is present. You will not know whether there has been separation, whether the DNA is there. So after you've completed the DNA, we now use gel electrophoresis to be able to visualize it. I want you guys to just see so that you don't. I don't want to be giving you abstract knowledge. That's why I'm still using video, so at least you know that this is how those things are being done. So after the PCR has completed, you just program it. It's like a programmable device. It is going to continue. Then the end result of the PCR is called the PCR product. Okay, so you are now going to load it onto gel. So you can see that this is blue, but the PCR product is colorless. So something has been added. It is called the loading dye. Because if you did not put anything to help you visualize the coloring, the thing will still continue to run, but you will not be able to see it. So that is why when you want to do gel electrophoresis, we had a loading dye to hit. And the loading dye is the one that gave the PCR product that blue coloration. Now, so at the end of the gel electrophoresis, it will have separated, but it's on the gel, so you may not be able to quickly visualize it. So this is why we have gel documentation system, in which you'll be able to see the patterns on the computer. You will now be able to print it out. So this is how you see if there has been separation, if there has been expansion. There is a particular, as you see, this is the thread that we now look at, the location, and now it has moved, whether from this point to a particular point. So in essence, so what I'm trying to say is for PCR, the difference, what we are doing in PCR is that we are mimicking the way through which the body, we are mimicking DNA replication, but we are restricting that DNA replication to a specific sequence, to a specific region, of the DNA, so that it will be the only one that is replicating. So how do we tell the machine, how do we tell the process that this is the only region that we want to replicate? There is something that is called primers. I won't go into the details of primers, but we make use of something that is called primers to specifically tell the DNA that this is the only region that we want you to amplify. So PCR mimics the biological process of DNA replication. So the goal of the PCR system is to produce more copies of a specific DNA sequence. You have 23 chromosomes, and all of these 20 chromosomes, each of these chromosomes, you have 23 pairs of chromosomes, which means you have 46 chromosomes. Each of the chromosomes contains thousands or hundreds of DNA. So, sorry, thousands or hundreds of genes. And each of these genes contains thousands or hundreds of thousands of nucleotides. But we are telling me that only a specific sequence of nucleotide we need more copies of it. So it will be the only region that is going to amplify, that we are going to have many copies. So that is what the PCR does. It enables us to amplify a specific region of the DNA, a specific DNA sequence. So, and with this, which simply means that if we know which part of the body, instead of using the DNA probe, you start transferring, put this on it so that if it hybridizes, we can now just go straight and say that that region of interest, if it is present in this patient, we are now going to amplify different regions. We are now going to see if it is now present. So our thinking now is much different than we were thinking when we were using RFLP, or when we're using what's this method again? Southern blotting. We are specifically amplifying known regions of the DNA to have more copies, and we are now going to match it. So PCR greatly amplifies the amount of a specific region of DNA. And in the PCR process, the DNA sample is denatured into the separate individual polynucleotide strand through eating. So it's through the process of heat that the DNA strand would, I'm not taking DNA, but that is what we use. So we use 2 oligonucleotide DNA primers. The primers are the ones that will tell the system which part of the DNA sequence we want to produce more copies of. And they are very much available now. And within 2 hours, you can actually get 1 million fold copies of that specific sequence. So which means this is the reason why even if it's just a tiny blood that was dropped at the site of the crime, that small sample is enough to be able to help us identify it, because if it's just 1 copy of the DNA that is present in that small sample, with the PCR, we can amplify it and produce thousands and millions of copies of that DNA and now match it. So what the PCR really does for us for now is that it amplifies a specific DNA sequence so that we have large copies of it. And we are now the one that will tell the system which sequence or which region of the DNA we want to amplify. So the way that is what we do. Now, how do we make sense of it? How do we apply this? Don't forget, everything we are talking about is still DNA fingerprinting. So how do we apply this in identifying? So there is something that is called STR analysis. It is called the short tandem repeat. I know you guys are already overloaded, but please stay stick with me, you are still going to understand it. I'm not trying to. So what we do is that STR stands for short tandem repeat. It is a very small region of the DNA. It's repetitive. It can be 4 base pairs. 4 base pairs simply means we have bases adenine, thymine, guanine, cytosine. So they are just 4 nucleotides and they are always repeating themselves. And so when they are present in, so everybody, so the difference is that because they are always repeated, what is different from one person to another is that me, you as a person, your own STR, you may have like 20 short tandem repeats, while another person would have 5 short tandem repeats. So the number of the short tandem repeats is specific to one person. So the short tandem repeat is a specific region of the DNA, very short, very small, but it's always repeating. But the number is specific from one person to another. So that is what I want you to take from there. The number of the short tandem repeats is specific from one person to another. So what we do is that we specifically target these regions and we calculate the number of short tandem repeats that the person has. So I can, like I said, I can have 20, while the cost rep would have 15. So which means if we have a sample that somebody found on the crime scene and we calculate the region, that short tandem repeat region, if my own is 20 and the cost rep's own is 15, and for the sample that is we found at the crime scene, we calculated the short tandem repeat and it is 15. Who do you think carried out the crime? Let me explain my question again. I said the number of short tandem repeats. Okay, yes, that's very good. I said the number of short tandem repeats is specific for person. For every person, you have a specific number of short tandem repeats. So I now said, for my own, as mu, for that region, the total number of short tandem repeats that I have is 14. While assistant cost rep has 8 short tandem repeats. My own is 14. The assistant cost rep's short tandem repeats is 8. So we now saw a sample at the crime scene. And after carrying out the short tandem repeat analysis, we found that there are just 8 short tandem repeats. So, Chibueze Nwankwo, who do you think performed the crime, conducted the crime? Chibueze Nwankwo.
So I got third cost rep.
Why? Defend that. Why is that so?
So I said everybody has unique short tandem repeats, and her own was 8, and the amount that was seen in the crime was 8.
Is that clear to everybody? If it's not clear to you, guys, I'm really trying to ensure that you have a strong background in molecular biology, because if you're able to get all of these things, you will never struggle to understand molecular biology. So if you don't understand what we've been talking about, please indicate, and I'm going to come back and explain to you. So let me give you 5 seconds now. The floor is open. If it's not clear to you so far, let me know.
Can you come again, like this last line?
Okay. So I said the short tandem repeat. So you've already gotten PCR. PCR allows us to amplify the specific region. We can amplify it. So for the short tandem repeat, it's a specific region of the DNA that is always repeating itself. There's a video I'm going to show you that tells us how it's done. But the expansion I wanted to get is that this is based on, we now said the number of short tandem repeat that everybody has is peculiar. That everybody has a peculiar number of short tandem repeats. That my number of short tandem repeats is different from your own. So when we calculate, so what we just do for us to be able to specifically identify who owns the sample is we analyze the short tandem repeats in the sample at the crime scene. Then we compare it with what each person has. So I now said, if my whole number of short tandem repeats for like ACG, STR is 40, and in the crime scene, it is 25. So automatically, I'm not the person that committed. It's not my own sample. So if another person now comes, and if they have like 20 suspected persons, we are going to have 20 different numbers of short tandem repeats. But the person that has the highest match with the sample, with the STR analysis that was found at the crime scene, is most likely the person that committed the crime, is most likely the person that has this number of short tandem repeats. So the number is used to determine what happens or who has the closest match to hit. So that is how it is. Like I said, is highly peculiar, and so each STR is polymorphic, and each STR allele will only be shared by around 5 to 20% individuals, and the pattern of alleles can identify an individual quite accurately. And so we have different STRs. So when we are now, it's not just one STR, there are several STRs. So let's say for us to increase the likelihood of a match, we can now target like 5 different STRs, or some people will go as far as 16. So for this location, I have 16. So it means when you are having like 16 specific results and there is a match, it is most likely that you are the person that the sample is. And don't forget, we still say that paternity too is used for paternity disputes too. If a father, if you are trying to fight over the paternity of a particular child, the Y chromosome of the baby will only be gotten from the dad. So what we only do in this case is that we look for STRs that are present on the Y chromosome that is peculiar to the father. So if 3 people are arguing over that, okay, this is my baby, 3 men are arguing over the fatherhood of a child, what they will simply do is that they look for STRs, such tandem repeats that are present on the Y chromosome. Then they are now going to match the STRs with the number of STRs with each of the fathers. So let's say there are 5 STRs on the Y chromosome. They are now going to analyze those 5 STRs. So for the one STR, if I have 20 copies and my baby has 20 copies, for the other STR, it has 5. So they are now going to look at the match. The one that is closest in match in the number of STRs to that of the baby is the person that is most likely to be the father of the child. So what we are trying to tell you, everything I'm trying to do today is to let you have a better understanding how we use genetic materials to make identifications and to make conclusions. So this video, I think, is really, now let me see if I can quickly, so that it breaks down the STR concept. And yeah, and I would like to share with you guys. Now, so I think it's loading now. It's somehow, but I think it will get the job done. I just want you guys to have an idea. You don't have to be hard on yourself, but at least you will never say that you've not heard about this concept before.
Hi guys, today's subject is about STR. Hi guys, today's subject is about STR analysis. Hi guys, today's subject is about STR analysis. STR analysis is a molecular diagnostic method which is very useful to compare specific loci on DNA from 2 or more samples. STR stands for short tandem repeat. STRs consist a little sequence of 3 to 7 base pairs, and then that sequence is repeated multiple times. So let's take an example of an STR which is called TH01. This STR involves the sequence ATG. The ATG sequence is repeated several times. The number of these repeats varies from individual to individual. STRs are available in the non-coding regions of the DNA. STR analysis measures the exact number of the repeated units. Each person has a unique number of repeats. A person is likely to have a similar number of repeats to...
So if you look at this picture, and you will see that there are different, like I said, there are different STRs. This is an STR in this region. This is another STR. So there are several STRs, and each person's STR is peculiar to it. The number of my STR1 is different from your own STR1, from the number of your own STR1, even though it's the same code, it's the same sequence. They are the same, but they are different in number. So, and there are several STRs.
Their close relatives, a person, repeated unit. Each person has a unique number of repeats. A person is likely to have a similar number of repeats to their close relatives. But people more distantly related to them and complete strangers will have a totally different number of repeats. In contrast to RFLP, which uses restriction enzymes to cut DNA, STR analysis uses probes which are specific for the desired regions on the DNA. And a PCR is used to determine the lengths of the STRs. A probe is a fragment of DNA which can be fluorescently labeled. The desired region is amplified by PCR, put on a gel and analyzed by capillary electrophoresis. The smaller pieces migrate faster than the larger pieces through the gel. Each little fragment is detected when it passes nearby a laser near the bottom of the gel. The electrophoresis is attached to a computer which detects the labeled fragments. The computer gives an output with peaks based on their sizes. Each peak is an allele and each locus occurs as a pair of peaks. One of the peaks comes from the mother and the other one from the father. If there is just one peak available, it means that the allele is homozygous and that the parents have the same number of repeats. If there are two peaks, it means that the allele is heterozygous and that the parents don't have the same number of repeats. There will be numbers available on the peaks. Those numbers gives the amount of repeats on a chromosome. So for example, this person has 12 repeats on one chromosome and 14 repeats on the other chromosome. On loci D7S820 you can see that the person has one peak, which means that he is homozygous for this allele. This method is used to make discrimination between unrelated individuals because they have different numbers of repeat units. This method can also be used to do a paternity and maternity test. So here we have an example. Each color stays for a different STR. At the top is the profile of the child. In the middle is the profile of the mother and the bottom is the profile of the father available. And you can see that within each pair of the STRs of the child, one of them comes from the mother and the other one comes from the father. If there isn't a match, then it might be not the true parent.
STR analysis.
If
So this is really important because so guys, so far, is this video clear? Like I said, don't be ashamed, don't be shy. If it's not clear, I can always explain. I know this thing, so you have nothing to worry about. So is this clear to you, or you need me to?
Sorry, it's not clear.
Okay, great. So let me look at. I think it's a tool that I can use to explain. This is what we do in STR. Like I said, it is very specific. So we have a particular. So this is take this as a DNA strand. Okay, there is a gene region. The gene code.
to have a similar number.
The gene codes for something. Why the STR is just a series of nucleotides. They don't code for anything. And that is why we said the STRs are found in the non-coding region of the DNA. They are not coding for any protein, but they are always there. And we said there are several STRs, short tandem repeats, and the number is specific. And so, which means this is STR1, this is STR2. They have their whole specific name, but just take this one as the first STR, the second STR. So for STR1, there will be, like I said, each person has a specific number of short tandem repeats. So I can have 20. You can have 5, for STR1. I can have 8 for STR2. You can have 4 for STR2. So, and it's not just for us to make an interpretation, for us to determine who is the true father of the child. It simply means we need samples from the father, we need samples from the mother, and we need samples from the child. Which brings us to this. So which means that I want to get to the, so which means we are going to amplify the specific region, the specific STRs using PCR. When we've amplified it, if you remember the gel electrophoresis that we said, so this is still another method of gel electrophoresis. But in this case, it is now coming, the result is coming out as a graph as peaks that gives us the total number of the STRs that we have. Now, let me use this sample now. This D8, this first one you are looking at, D8S1179. Let's take it as STR1. When you have these two peaks, it simply means you are heterozygous because, okay, don't forget, these things are regions on the DNA. The DNA are on chromosomes, and you are inheriting your chromosomes from two of your parents. You have one on chromosome, one chromosome from your dad, another chromosome from your mom. So when this is happening, it simply means that from your dad, though, from the chromosome 4 for your dad, the STR1 on that chromosome from your dad only has 12 numbers of STR, while the one from your mom has 14. The numbers are different, but you have both because you are from both parents. So, and you have 2 copies of each chromosome. So I'm trying to say that your total chromosomes, you have 23 chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosomes. So I now said for the STR analysis of chromosome 1, it is possible for you to have different numbers of STR from both parents. So as you can see, this one is 12, that one is 14. It simply means from the copy you inherited, you got from your dad, there are 12 STRs, or there are 12 repeats of D8 of STR1, while your mom has 14. For this second one, D21S11, your dad's chromosome has 28 STRs, while your mom has 30. While for this 7X820, both of them have the same number of STRs. That is why we are having a single copy. And which means you are homozygous, they are the same. And while for CSF1P0, this is also homozygous. They both contain the same number, which is 10. The person that asked the question, is it clear so far?
Yes, sir.
Now, let me now continue how we make that interpretation now. So please look at the peaks. These peaks are very, very important. Because when we now want to make interpretation on who is the father or who is the mother, whether they are true fathers or not. Now, this is how it's called. We have the pattern for the child, that the paternity is in dispute. We have the paternity, we have the platform, we have the results for the mom too. If you remember, we said this one will be like 12. The peak is what is used to calculate it. They can be 12, this can be 11, and the one from the suspected father.
We have an example.
So when
Each color
so don't forget, so what we need to do is that this is the child, which means at one point it must match that of the dad, of the mother, because there are 2 copies of that chromosome. As you can see, each color represents a particular STR on a specific chromosome. So if you are getting 2 copies from your dad, it simply means everything must match that of your dad from your mom. Look at the first purple, the first one for the child. This first one is matching that of the mother, while the second one is matching that of the father. As you can see, the mother's STR for the first one is also different, because the other arm, the mother got, which simply means this is the chromosome, because you are going to inherit 1 copy of the 2 copies of your mother chromosome, and your dad is also going to give you 1. So this is one that matched with the dad, with the child, and this is the one that matched with the mom, with the father. So this is how the child got his own different assortment of the STR differently. Now for the other SCR2, you can see the first one too. The first one perfectly matches that of the mother, while the second one perfectly matches these of the father. So we can still see that there is alignment. So I'm just trying to tell you how they do paternity testing, so that you'll be able to figure it out on your own. That is how they do STR. You now still go again to another STR3. STR3, which simply means this first copy was gotten from the father, while this copy was gotten from the mother. Even though both of them, the father and the mother, they also have different STRs, but it is always passed down to the child, depending on the chromosome that the baby has. Now, let's look at a case where this child, in this case, the father is the true father. As you can see, everything is matching. For the one that is having homozygous, that is always the same copy. It simply means that even though the father and the mother, they have different chromosome too, they still have something that they are shared in common. The baby has one, and there is one on this location. There is the peak on this location. There is another peak on this location. So which means it's still possible that the baby got chromosome, the first chromosome and the first chromosome from the daddy, and they're exactly the same. That is why it's there. While for this one, as you can see, this is the one that the baby has from the father. It's got the first copy is from the father, the second copy is from the mother. So you're able to perfectly align it as true picture. But in a case where this is not happening like this, you can say that, mother, I'm not sure this is the father, this is the true father of the child. So when you have different fathers like that, different claims to a child, this is the method that they use. You bring about different STRs. So the person that has that question, don't be shy. Is it clear?
Yes, it's clear. I have one question.
Okay.
When you were explaining, you said we have one pair of sex chromosomes and 23 pairs of what?
No, I did not say that too. So normal person have a total number of 46 chromosomes. 46 chromosomes, that is everything that you have. So that 46 chromosomes are now broken into 23. 22 for autosomes, chromosomes 1 to 22. They are body chromosomes. They are the same, irrespective of your gender, whether you are a male or a female. We both share the same chromosomes 1 to 22. So you now have two pairs of that chromosomes 1 to 22. That is 44. For the remaining two chromosomes, they are the sex chromosomes. For a female, you're expected to have two X chromosomes. So two X chromosomes plus 44 body chromosomes, or the first 44 chromosomes. That makes it 46. While for the males, you're expected to have one X chromosome and one Y chromosome. So that is you have two sex chromosomes and 44 autosomes. Is that clear?
Yes, sir.
So this is how we okay.
Sir, I have a question on that paternity test. Sir, what if the fathers that should be tested, what if they are twins or they are brothers? Won't it be more difficult to know who is the true father?
It will not be.
Since those fathers have the same parents and they have similarities too. Thank you.
That's a good question. And the only situation where we might have problem is if they are genetically identical. And even at that, again, there will still be room for chance, because don't forget, whether you are getting, look at this pattern that you have. For the first, let's say the one that is in blue, the child got the first chromosome from the mother and the second chromosome from the father. In the one that is, now look at, It got the first chromosome from the father, the second chromosome from the mother. Now look at the one that is in red. The first chromosome is from the father, not the mother this time. While the second chromosome is from the father, is from the mother. So even if they are genetically identical, the profile, the STR analysis of the child will still be different, because if in Yorubas we say we have Taiwan and Kende. If the child is getting the first chromosome for the blue chromosome from Taiwo, it may decide that for its own second STR, it is going to pick the second first. I don't know whether it's random. Even though you have the same mixture, the one you can pick first for each round may be different. You may pick A this time and you pick B that time. So even though they are genetically identical, the fact that it is picking the first this time, the second that time, will still bring about that variation. I don't know whether that makes sense to you.
Yes, sir.
So who else has another question for me? I just want you guys to see how this paternity thing is scientifically done. And
Sir,
okay,
listen, my question is not based on the paternity testing stuff. The probe that you mentioned, I was a bit confused, because I'm like, are we the one to prepare the probe, or the probe is already standard? It's just for us to pick and connect with the forensic,
yeah, that's a very good question, and thanks for pointing it out. Yeah, many of these probes are commercially prepared, because already all the things that you are looking at now, we've already identified the STRs of interest. So the probes for each of these have already been designed. So they are largely commercially available. We know the target for this probes. We just want to see the pattern. How many copies do you have? Because they are really, really specific in the sequence. And so they are specific. So we already know them. So they are commercially prepared, and you are just introducing it. Is that clear? So, but sometimes if you are really good, if you really want to go, you may decide to prepare your home probes. For instance, if it's for research purpose, and you're trying to test it in the population, you can prepare your home probe. Because the sequence is always specific. I remember when I was doing my own lab work for my PhD, I tried to design my own probe. I told them the sequences that I want in the probe, and I sent it to a lab in France. They produced it and they sent it to me. I was able to use it. So if you want to prepare it yourself, it's available. You can do that. And if you want to buy commercially prepared probes, they are also available. Exactly.
Okay, sir.
Who else has any other question for me? Now, so this is how they do the result paternity testing. And also if it's for a crime, if you are for something that you found at a crime scene, this is not a case of father-mother. So they just delete the mother, because we are expecting it to have a perfect match. Now look at the child. Look at the child as the reference sample. If it's for something you found at a crime scene, that is the reference sample. So this is suspect A, suspect B. So for us to know that this reference sample is from me, when you do the STR analysis of this reference sample and my own sample, for us to conclude that I'm the person whose sample was found at the crime scene, there must be a perfect match. It must be perfect. The same peaks that you are seeing must also be seen in my sample. Since it's from you, we are saying that it's from me. So it must be a perfect match if we are using it for forensic investigation. And that is how we are able to say that this sample is from you, that sample is from you. So all those CSI films that you are watching, all those action movies, police movies, investigators movie that you are seeing, that is how they make their whole conclusion. That is how they infer, that is how they know who the criminal is, and that is how they know whose sample was found at the crime scene. So when you identify, on your screen, you are looking at just like 5 STRs. But ideally, for you to be able to convincingly say that this is the person that perpetrated, that committed a crime. Sometimes you may have to conduct up to like 20, analyze up to like 20 STRs. And when you are having a perfect STR analysis for 20 STRs, you can convincingly have a more robust result and say that we have 90% possibility, 90% chance that this baby belongs to this father or this baby belongs to this mother. So the first thing they just do is that they figure out the patterns for the mother. So they figure out which ones are for the mother. It quickly matches, it quickly aligns. Then the father is one that will now be placed. And interestingly too, there are softwares too that would automatically do this for you, and will tell you which one is this coming from. So that is what I think is really, really important. That is how short term, then repeat, analyzed. And is there a question so far on this before I continue? So like this, continue, say whatever you want to say, please. Okay, so that is STR for you. Now, there is also Y chromosome analysis. So when we are trying to, for Y chromosome analysis, we know that all things, ceteris paribus, all things being equal, a child is expected to get the Y chromosome only from the father, because women don't have Y chromosome. So if you are also trying to do some forms of paternity testing, you may have to also do Y chromosome analysis. And this is due to the fact that, so we have Y STRs. When you see Y STR, it simply means the short tandem repeat on the Y chromosome. But so they are paternally, like I said, the STRs are paternally inherited. The STRs are paternally inherited. They are gotten from the father. And so when you have that, you really have to now say that, okay, this is where the father is, this is where the sample is from, and allows you to get that. But there is a major problem with relying. You cannot rely on this alone, because when you look at the Y chromosome, it's very small, and so it may not be able to provide individual identification alone. For instance, if you want to identify an individual, if you want to say that this is a person that has this or not, it may not, relying on Y chromosome analysis alone may not be enough. But what Y chromosome analysis can tell you is that whether this sample is from a male or female. For instance, if we are suspecting two persons, even though we can be excited to say, oh, this is a paternity dispute, let's use Y chromosome analysis. But the Y chromosome is really, really similar in many males, as long as you're male. But if you are trying to delineate, or if you are trying to identify which gender has this problem, because when you see a blood sample, when you see an hair strand, when you see a saliva, when you see a body fluid, you may not be sure of which gender, whether it's from a male or female. So in this case, the Y chromosome analysis tells you what it's doing. So for the person that asked what this class topic is, I think this is the title of the class. It is called DNA profiling. Another name for DNA profiling is DNA fingerprinting. So that is what we do in Y chromosome analysis. It serves the purpose of selecting, resolving a missed DNA sample, if it's a male or female. So if you have a blood sample and you have suspects already, and you do Y chromosome analysis and it turns out positive, so you can just exclude the females, that you ladies be going. It has nothing to do with you. This is a sample from a male. Then after you've eliminated that on the gender, if you had like 5 suspects and 3 of the suspects are females, then you can actually now exclude the 3 and said, okay, we have 2 males, so it is most likely to be between 2 of you. But if you see the area that I've highlighted in red, analysis of Y chromosome yields weaker results than autosomal chromosome analysis with regarding to identifying individuals. So you now still need to do autosomal STRs, other STRs on other body chromosomes for you to be able to do it. And the reason is this: the Y chromosome is like the smallest chromosome, so it has very few STRs. And if it has like 1 or 2 STRs, you may not be able to establish that individual variation. So that is why we still don't look at it, but it can tell you the gender of the person. Then that is for the paternal side. If you want to look at the maternal side too, you can look at what is called the mitochondrial analysis, mitochondrial DNA analysis, because it's said that if you want to, mitochondrial DNA are inherited from the mother. So the DNA of the mitochondria, the mitochondrion is an organelle in the cell, and it also contains its own DNA. So if there is a missing person, and for instance, if there is a missing person, or you still don't understand, you want to understand whether these 2 persons are related. If they share the same mother, look at Africa, for instance. In Africa, we have men that trying to draw a family tree in Africa is very, very difficult, because our forefathers are polygamous in nature, which means we can have a family that has a father that has married like 20 wives. Now imagine trying to draw a family tree with 20 wives. It will be extremely, extremely difficult. So this is the reason why when we are trying to draw a family tree from that, it's better to draw it using the mother. The mother serves as a definitive purpose, because the mitochondrial DNA is passed, your mitochondrial DNA was gotten from your mom. So which means, and our own was gotten from our home mom too. So your grandmother, which means you share the same mitochondrial DNA with your great grandma from your mother's side. So for you to draw your clear family tree, we can actually get your mitochondrial DNA. So if we are not sure about the identity of the lineage of somebody, if we are not sure about where somebody is coming from, then they can actually use the mitochondrial DNA to use as match reference. So that is how this is done. So if somebody is telling you that I'm connected to your mother, or your grandmother has like 3-4 children, I want to really prove that you are sharing the same maternal lineage, you can compare a similar analysis for the mitochondrial DNA. When somebody comes out and says he's a royal princess, then you want to disprove it. Mitochondrial DNA can also be carried out. Another thing that okay, what's the question?
So I have a question. Concerning that Y chromosomal testing, I don't know. I feel it's actually restricted to only males when it comes to parental testing. Because if it's a female child, there's no Y chromosome present now. How they want to know?
That's a good question. So if you remember what I said, I did not say they don't use Y chromosome to resolve paternity disputes. Even though it's obvious, okay, this is a male child. Wow, yes. So let's compare the Y chromosome from this person to another. The reason is that, like I said, the Y chromosome is a very small chromosome. The Y chromosome is very, very small in size. And when you are looking at something that is very, very small in size, the reliability for you to be able to rely on the data, you must have sufficient data. So the only purpose for now that Y STRs, Y chromosome short tandem repeats can help us with is for us to be able to separate whether a sample is from a male or female. That is the best application for Y chromosome STR because it does not have sufficient number of STRs for us to rely on it. It's a very small chromosome. I don't know, chromosomes. So I want you to see if there's a picture. If there's a, I'm trying to see if I can show you a picture of, yes. Okay, I think I've seen a picture of the Y chromosomes, of the chromosome so that you can, like I said, you guys are just starting your journey. It's better you get everything clear. So there is this picture on Wikipedia that I think answered that question. So everybody look up, please. Look at the chromosome. If you are, the largest chromosome is chromosome 1. So as you are going down to chromosome 22, the size reduces. Can you see? Chromosome 1, look at how big chromosome 1 is. Look at the 2 copies. You have 2 copies of chromosome 1. Chromosome 2 is smaller than chromosome 1. Chromosome 3 is smaller than chromosome 2. But now, as you can see, as chromosome 18 is reducing in size, chromosome 22 is also is like the smallest autosome. But now look at chromosome, this is chromosome X. And look at chromosome Y. Chromosome Y is very, very small. The number of STRs that you are going to see on chromosome 1, it will be so many. Compare it to the Y chromosome. It will be so few. So let's say on chromosome 1, you can have like 45 STRs. Chromosome 4, 25 STRs. But chromosome Y, 3 STRs. So you cannot rely on the variations on just 3 STRs with something that will give you like 45 STRs, 30 STRs, or 15 STRs. Does that make sense to you now?
Yes, sir.
So chromosome Y is very, very small. But the only application, the major reliable application that we have for it is determining whether a sample is from a male or a female. So when you do it, when you don't know whether, when you have multiple suspects of both gender, you don't know whether they are male or female, the first thing you just need to do is that, okay, let's do Y STR analysis. If there is a match, if you have Y STR analysis, it simply means that you are a male. So you can just say all the females start going home. And you now focus on the males. When the males are now here, you now do STR from autosomes, from chromosomes 1 to 22. Does that make sense now?
Yes, sir. So we can basically say this Y chromosome analysis is just to separate, like it's just to know whether the suspect was a male or female. So not basically used in paternity test actually.
That's very correct. So that is how we can apply it. So it does not serve paternity dispute because in paternity disputes, you will say that, are you 100% sure? If you've seen a paternity, let me see if I can see a paternity test result. Example. I want to see if I can. So in many cases, they will say that it has like, I want to see if I can see a paternity test result to show you how it looks like.
So we have another class.
Oh, you have another class. Hey, yeah. So it is going to tell you the likelihood that this person is a father, or likelihood that this person is a mother. So, and the ratio. So the more the paternity, the more the STRs that you've analyzed, the larger the reliability of your results. So, so look at this. So look at this. So look at the number of locus. These are the STRs that, look at these are the STRs. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. Okay, now they've tested 21, and they are looking at, can you see the STLs for this one? The baby has 13, 15, 13, 15. If there is match, this one is 17, 18, this one is 18, 20. There can be slight variations because the baby is still, now, in the result, probability of paternity. So it means that this man has a 99.9998% chance of being the father. So if there's another confusing father that is saying that the baby belongs to him, they will say that maybe that one is not like 40%, 40 point some 40.55 something. So the one that has the higher probability is most